then in the cytoplasm we do glycolysis. Glucose comes in. We invest a little bit of energy, but we end up netting 2 ATPs. We also net 2 NADHs, which, remember, the reduced NAD carries electrons, in this case, into the mitochondria and then over to the electron transport chain, which is the topic of this section. The 2-pyruvate that are made as a result of glycolysis get imported into the mitochondria. When it comes through, we lose a carbon dioxide, and now the 2 carbons are preserved in acetyl-CoA, which drops off the 2 carbons into the citric acid cycle. We went through that. The products of the citric acid cycle are 2 ATP. These are both made by substrate level phosphorylation, meaning an enzyme moves the phosphate and then therefore harnesses the energy and transfers it to the ATP molecule directly. Now we're going to jump into the more production style method of making ATP, almost factory-like. We're going to take the NADHs that are produced along the way, with the most coming from the citric acid cycle, to FADH2. We'll see the difference between these 2 carrier molecules and how they drop off their energy here and make a boatload of ATP. Remember, this is happening inside the mitochondria, and then we're going to talk about stuff that happens on this inner membrane. This line is an inner membrane. Focus it. Okay, so the electron transport chain, next step. Before we get to how it looks in the membrane, let's talk a little bit about energy. Remember, free energy again. So relative to oxygen, which we call the final electron acceptor, it's at the lowest energy level. This is the reason why we breathe. It's the end of the electron transport chain. Relative to oxygen, we have these other molecules that are embedded in the inner membrane of the mitochondria. Some of them are transmembrane, some of them are peripheral to the membrane, and then there's this collection of other molecules that get oxidized and reduced. Some of them share a name called cytochrome. Do you have to know the names of all of these? Absolutely not. I'm going to mention them simply because they have names. And the cytochromes are highly conserved through evolution. A topic we're going to come back to again and again through the year. If a protein is critical for survival, it's been preserved. You can trace its amino acid sequence back and then even its DNA sequence back and find that there are very few changes. So we can show how things are evolutionarily related to each other by looking at sequences of proteins. Cytochromes are favorites to do this analysis on because everything that evolved to survive in the presence of oxygen, we'll get into a lot of that later on, have basically used some form of this chain to generate their energy. Okay, back to what I was talking about. So there's a couple main complexes that we'll talk about how they pump hydrogens through. But right now let's focus on NADH is going to arrive at one side of the membrane from the matrix after doing the citric acid cycle and such, and drop off its energy, basically drop off the electrons. And remember, when an electron gets transferred to something new, it gets reduced. So NADH will lose its electrons and get oxidized, and its thing that it's handing it off to, in this case the FMN, flavin mononucleotide structure, is going to accept that energy in the form of electrons and get reduced. It will then exchange its energy down this chain to something that is lower and lower and lower free energy, is more likely to get reduced all along. And that's why they go down this nice little chain. Okay, so there you go, energy. You know what, I'm going to jump ahead over here for a second. So those are the guys that we just drew. They color-coded them the same, this purple. We're going to come back to ATP synthase in just a minute. Let's focus on transferring that energy through the membrane. So again, color-coded to hopefully help us out a little bit. This is now the inside of the mitochondria, the matrix. This is where the citric acid cycle took place, and we produced our NADH conveniently on this side of the membrane. This orange area is actually the inner membrane space, the space between the bilayer of those folds, the cristae that fold in the mitochondria, and the exterior of the mitochondria you can think is up here. So we're going to drop off our energy in the form of NADH getting oxidized and transfer its electrons to this first complex. Every time you transfer that energy, we need to do something with it. And what we're going to do is pump hydrogens across. This is a transmembrane protein that has very specific specificity, I should say, for hydrogen ions. Hydrogen ions get pumped through when that NAD drops its energy off here. This complex then reduces quinone. This is an interesting one. It's one of the only ones that is a lipid-soluble molecule. It floats between these two, and I'll show you an animation that's going to make this come to life a little bit. Energy transfers from here to here because this guy wants to be reduced, and then the quinone is going to transfer its energy over to this next complex. Again, we want to do some work when we're transferring the energy, and we pump a hydrogen across again. That energy gets transferred then to cytochrome C, which is a peripheral protein, kind of tethered to that inside of the membrane here. And then it transfers its energy to this last one, which also pumps a hydrogen. So focusing on NADH, it has enough energy in it when it transfers it to these complexes to pump a total of 3 hydrogen ions. Is this sounding a little bit like active transport that we talked about in the other chapters? We are using energy to do some work. Basically, we're going to accumulate a gradient of hydrogens, and those hydrogens are going to pass down this complex, which I have to go back to the other picture again, called ATP synthase. This is a multi-subunit mechanical enzyme that literally turns in the membrane. They have models that show this. And when it turns, the enzyme attaches a phosphate to an ADP and makes ATP. So it's a very mechanized enzyme. Okay, I focused on NADH here. Now, remember, we keep bringing up this FADH2 guy. So he's worth a little less energy than the NADH. It drops its electron off at the quinone, so it bypasses this complex. So it can only have enough energy when it reduces all these guys to pump 2 hydrogens across. That's why it's worth a little less energy. So NADs can potentially make about 3 ATP, because for about, and I say about because it's always sort of a relative term, every hydrogen that goes through makes an ATP. So if this guy drops off its energy and pumps 3 hydrogens, we make 3 ATP. This guy drops off his energy, he pumps 2 hydrogens, we make 2 ATP. So in this picture we're making 5 ATP. Let me go backwards and show you that structure. So the ATP synthase is a phenomenally complex enzyme. We're embedded in the membrane again. This is the matrix. This is where the Krebs or citric acid cycle took place. This is that inner membrane space where we've been pumping those hydrogens. So they know that the hydrogens are attracted to this little pore, and the hydrogens bind to proteins that are subunits that are in this thing they call the rotor, subunits that make a wheel, and it electrostatically turns that. Remember, positive charges are not going to be very happy in this non-polar environment. So there's an interaction that kind of tweaks it. And then a previous one, after it goes all the way around, goes through that pore and out. And that forces, again, the revolution of this. And then there's a complex enzyme reaction down here that, again, transfers the phosphate to the ATP. Phenomenally complex method of making ATP, but really cool because it's like almost a machine. Crank, 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 make ATP every time you move the hydrogens through. So we have him in this picture here. So let's just put that into perspective one more time. So we were talking about the electron transport chain, which is embedded in this. See that they keep that orange? This is where the hydrogen ions collect. This is where our transmembrane transporter proteins are that receive their energy from NADH, pump the hydrogens across into here. Hydrogens collect and then go down that ATP synthase. Okay, so that's if, I'm going to go back one more time. That's if we have oxygen. So as long as oxygen is present in the system, you are actively breathing. The earthworm is getting oxygen across the membranes of its skin. The fish have their gills working properly, and oxygen is being loaded into the bloodstream, transferred to the tissues that need it, and so on. We'll go through all that a lot later. If oxygen is in the system, the whole thing works great. We always are pulling this electron down to its lowest energy level to reduce that oxygen to make a water molecule. What if you don't have oxygen around? You hold your breath, you can't breathe anymore, something happens to your diaphragm, you're not breathing. You take the earthworm and you make his skin dried out, so now he can't transfer the oxygen across the membranes, whatever. Everything backs up and it stops. If it's in an anaerobic environment, no oxygen environment. You could also have a poison that binds to one of these and stops up the system. Carbon monoxide will do that for you. Cyanide will do that for you. A bunch of other nasty things, poisons will do that for you. And basically you stop all this. Or what if you're exercising like crazy? You're one of these cross-country runners that run like crazy and go to oxygen debt. You've used up all of your oxygen that's floating around and you're screaming, but your muscles still have to keep going. What do? You do fermentation. So here's glucose comes in, goes through glycolysis. That happens without any oxygen. What do you make? 2 ATP. Not a lot, but you can survive. But what you need to do for this cycle to keep going is to regenerate that NAD. Remember, there were a couple steps in that energy payoff phase where we made NAD go to NADH. We need to keep recycling the NAD to keep that whole system running. So we need a way to regenerate that. And there's 2 ways you can do it. If you're yeast, for example, and you are in the absence of oxygen, you go down a pathway to make ethanol. If you're a human, a mammal, and for just a little while, your muscles can make lactic acid or lactate, and then still make a little bit of NAD, go back and have that whole system keep going. So here are those 2 pictures. So here's alcohol fermentation. This is what yeast do when they're deprived of oxygen. Again, they do glycolysis, make those 2 ATP. Happy, you got a little energy. Now we're left with pyruvate. So what they do is they have an enzyme that rips off 2 carbon dioxides, and now you have what's called acetaldehyde. Don't worry about those names so much. And then another enzyme is going to come into play here. So we have 2 enzymes. And this enzyme has the ability to take your NADH and transfer its energy over to that acetaldehyde molecule to make ethanol. Ethanol is simply 2 carbons, those 2 hydrogens hanging off that carbon, CH3, methyl group over here, and then an OH over here. And then that builds up. But now you had some NAD, and it can go back and run glycolysis, and you can have a little bit of ATP to run. This is what yeast do when they are fermenting and making the CO2 or the bubbles in your bread. This is what yeast do when they are fermenting and make the bubbles that are in champagne, beer. If it's in wine, you let the bubbles go off. You don't want that. You just want to capture the ethanol, and so on. So in mammals, we don't make a lot of CO2, and we don't collect that ethanol because we know that that's actually a poison in our system if we collect it. And we make lactic acid, which is in and of itself is not that great, but we have a system in our bodies to clear away the lactate and deliver it to the liver to get dealt with, basically, and clear it out. So again, we run glycolysis. We get a couple of ATP to make the muscles barely go. Make pyruvate. And in our case, it's a 1-step situation where we take the NADH, grab the energy out of it, make lactate, and go back and run again. Zoom in. Goodbye. All right, that's about the end of that discussion. So if you have oxygen, you go into the mitochondria and make boatloads of ATP. No oxygen present, anaerobic conditions. If you're a yeast, to make ethanol. If you're a mammalian cell, you're going to make some lactic acid. So let's just talk a little bit about the control of the system. So we know that there's other food we eat and other sources for energy. The main source for getting energy is for glucose to go through glycolysis, to make pyruvate, to go through acetyl-CoA, go into citric acid cycle. Oxidative phosphorylation is electron transport chain production of ATP. So we know we need that. Can you get energy out of other things? What if you stopped eating? Don't have any more sugar in your system? Well, we can break down the fats in our body. Remember, a fat is a glycerol molecule with 3 fatty acids attached to it. Remember, fatty acids are a 18 to 22 carbon long chain. So if acetyl-CoA is 2 carbons attached to a coenzyme A that feeds into the citric acid cycle, which makes a whole lot of NADH to go make ATP. If you have 3 times, say, 20 carbons, so we have 60 carbons in 1 fat in those 3 fatty acid tails. They can be lopped off 2 carbons at a time. Here's your 2 carbons. They feed right into the citric acid cycle. You get lots of energy. For animals, we store our energy as fat for that reason. It's very mobile, it's nice and gooey, we can move it around, easy to carry around, and we can get a ton of energy from it. On the other end of the spectrum, proteins, not so much energy because you have, remember, each amino acid is a NCC, an amino group, a central carbon, and then a carboxy. And those can basically get chopped off and fed into this pathway with a couple of enzyme steps. Not very efficient. Proteins are usually doing some really important work in your body. It's not really the way we store energy, but last ditch effort, when all the fat's gone, you start eating up your